Oh, this is a better picture. Camels. Okay, camels originally come from America. Here, America. And then they migrate into South America to become Lima. And then they cross the so-called Pacific Bridge. Here, originally, the Alaska is connected to this Asia with there's a physical land connected between the two in here. But after so many years, that cross Pacific Bridge is broken. But before it is broken, camel migrate to Asia and Africa. That's why you have camel in Asia and camel in Africa. They all came from America. And then the fifth evidence for macroevolution is to do DNA analysis. And I personally consider that as the most objective, most accurate way to do, we call it classification, and also to trace the lineage of different animal. The closer for animals that share the same common ancestor, their DNA will become very similar. And the closer the two animals they are related to each other, or the closer their evolutionary lineage, the more DNA bases they will share between each other. For example, a human being and chimpanzee, they're cousins. They share a common ancestor. So they share a lot of DNA with each other. But let's say human and dogs. Well, dogs and human, we are also mammals, but we are not closely related to dogs. So we do share a lot of genes with dogs, and but we also have some genes that are not commonly shared. But when we compare human being with, let's say, a fish, then you see, wow, a lot of gene, a lot of DNA, they are totally different because we don't share a common ancestor. What is gene? Gene, they cook for specific trait or protein. We call it gene. Random sequences, they are the DNA sequence between genes, not fully no protein coding. Well, personally, I think that you may call the gene is called exon. And the random sequence we can call it intron. If you remember the beginning of the semester, we talked about transcription translation, okay? So transcription is basically converting DNA into messenger RNA. After we got the messenger RNA, we need to do something called splicing or some kind of modification. Basically, during this splicing process, we will remove intron, which is this non-coding region of the DNA of the RNA, from the exon. So we will keep all the exon because exon, they are in the messenger RNA that is for coding. That is for coding the protein. So this is the mRNA. This is also in the mRNA. So before the messenger RNA go to the cytoplasm, all the intron in the messenger RNA will be removed, and all the exons in the messenger RNA will remain, will be kept, and then when messenger RNA leaves the nucleus to start the translation process, then the messenger RNA contains only exon. What about intron? What do they do? Intron, one of the functions of intron is to stop the translation process. Yeah, the intron, they are recycled to stop the translation process. And as you can, but here what they refer to is DNA. Okay, they are referring to DNA. In DNA, they have the part that cook for protein. They have part that is not cooked for protein. So you may assume that the part of the DNA that will be cooked for protein is the part that will make exon on messenger RNA. The part of the DNA that is not cooked for protein, they will be the part that is cooked for the intron on the messenger RNA. So we are comparing the DNA sequence between human and other organisms. You can see chimpanzee, they are highly so-called conserved with human being. So the gene code for the protein is almost 100% identical in human and chimpanzee, but the random segment between gene is 98% similar. And as you go down, okay, dog is also mammal, okay, and then mouse is also mammal. You can see that even mouse, it is a pest, it is still 99% so-called conserved between mouse and human being. But when you go to reptile, chicken, you drop to 75%. When you go to flies, which is insect, 60%. Roundworm, they are even lower, 35%. That's why all mammals, we share the same common ancestor. That's because we have a high we can call it consistency, or the highly conserved DNA with each other in the mammals. Sorry, I forgot to mention. Yes, it's highly conserved between each other, but the differences is in the non-coding part of the DNA. The non-coding part of DNA set us apart from, let's say, from dog and chimpanzee. Yeah. The sixth evidence of macroevolution is the biochemical analysis. We compare protein, okay? So we compare DNA sequence, now we compare protein by biochemical or immunological methods. An example is this cytochrome C. Cytochrome C is a protein that commonly occurs in all animals because cytochrome C is one of the protein. You find it in the electron transport chain during cellular respiration. That's why as long as you're a living animal, you basically have cytochrome C. Because as long as you are a living animal, you will do cellular respiration to make ATP. When you do cellular respiration to make ATP, then you will have electron transport chain. When you have electron transport chain, then you have cytochrome C. Because cytochrome C is one of the protein you can find in the electron transport chain. So you can see that, okay, between chimpanzee and rhesus is the monkey living along the bank of rhesus river. So the rhesus monkey. You can see that, okay, they're highly conserved the amino acid sequence. This is what we call the amino acid sequence. They are highly conserved. You see the red one here, the red one in the rhesus monkey. It is different from chimpanzee here. So it's like, okay, for the 104 amino acids that make cytochrome C, only one is different between one amino acid difference. One amino acid difference between chimpanzee and rhesus monkey. But as you go down, when you compare whale, turtle, bullfrog, tunnerfish, and lamprey, as you go as you deviate from the same. Oh, sorry. Rat is different from human being. So chimpanzee is basically identical. The cytochrome C is identical to a human being. Rhesus monkey only has one amino acid different from human being. But as you go down, when you deviate from mammal, lamprey is a very primitive vertebrate. It's like a fish. Then you will have more and more amino acids that are different from human being in the cytochrome C. So this is the result of comparing the cytochrome C with different animals from yeast, mole, fish, turtle, duck, pig, monkey, and human. Even yeast, they are living organisms, so they have cytochrome C for cellular respiration. They have, at least 50 of the amino acid that is different from human being in cytochrome C. So fish you have approximately 20 different amino acids different from human being. So we show you 6 evidence for macroevolution so far. You need to know all 6, unfortunately, yes.